The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
So the little myosin, and there's another kind of close-up view of how it happens, but everything sort of is like being rowed across one another. So that's how the sarcomere contracts. And let's see, what's this one? Okay, we don't need that one quite yet. Myofilament. Okay, here's the little, okay, maybe this, well actually, no, let's do, there was one in here that had calcium in it. No, that's not the one I want. Come on, Dave, figure this out. Right down, it doesn't look like the one I want. That's not it. Yeah, I don't know. It's not going to start this one. Can't find the one that had calcium. I'm sure I'll find it.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
But now we got to get calcium into play here. Let's do this one, see what this one does.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So the initial important step here is when the little green thing here, which is calcium, binds to the troponin, causing the tropomyosin to move out of the way and expose these binding sites. That's why calcium is important, because it binds the troponin and then exposes the myosin binding sites on the actin. So that explains a bit of the role of calcium. And then we'll do this one, which is this thing about an action potential in the T-tubules. And this will,
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubular membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
They definitely play a role. Okay, one more. This is,
Professor?
yeah.
So it's saying the T-tubules, they allow for the action potential to go to
Yes.
the sarcomere to release the calcium?
The T-tubules, if you look at that model, like this back of you, what happens is the action potential is propagated into the muscle cell along the T-tubules. But remember, the T-tubules sort of cross right in that area of the sarcoplasmic reticulum called the terminal cisternae. So the action potential causes release of calcium from the sarcoplasmic reticulum, from the terminal cisternae of the sarcoplasmic reticulum, and that allows then the calcium binds with the troponin changes shape, the tropomyosin moves, bingo, we got muscle contraction.
So is there more like, so what we have to know about the T-tubules, they're just so the action potential can travel along?
It's basically the pathway for the action potential into the muscles.
Okay.
And there's one more thing on here that I'll show you, and this has to do with the neurotransmitters, the acetylcholine, and what happens at the neuromuscular junction. So let's see that one. We've done that one. There it is. Okay. So this is the last one, and then this shows you, and I'll have to talk about this in just a minute anyway, so it won't.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So a lot of physiology there, but the basic idea is the action potential, this is a neuromuscular junction. The action potential causes acetylcholine, which is a neurotransmitter, to be released. The voltage-gated channels, that's what you get in physiology, so we don't need to do it here. But the acetylcholine diffuses across this very tiny space and eventually causes, as they call it, a threshold to be reached on the membrane of the muscle, and that sort of thing they showed you spreading along this membrane is basically the action potential, which will go into the T-tubules and cause calcium release and that sort of stuff. Now, as I mentioned yesterday, and this can explain it a little bit better, it's not something we're going to test on or you really kind of need to know for anatomy, but it's an interesting concept. If we fill this space with something that's going to block the release of acetylcholine, then we can prevent muscle contraction. And what we can fill the space with is botulism toxin, Botox, in a very diluted form. So it, in essence, fills this space, prevents these voltage-gated channels from opening. Basically, it prevents in a simple way, prevents the muscle from receiving the stimulation to contract. And what wrinkles are basically are muscle contraction. So if we block that, then you don't get temporarily anyway, your wrinkles go away. So brilliant idea. So anyway, a lot of physiology in this one, but you get the idea. So those are the those are little videos. So we can get